we're 1 delta, and we investigated the single deletion of Escherichia coli K30 group 1 capsule biosynthesis system component, WZB, and found that it's not sufficient to convert capsule-independent resistance to erythromycin. So bacterial capsule is this thick layer on the outside of the bacteria, and one of its functions is to confer antibiotic resistance to contribute to cell survival. So for E. coli K30, the capsule formation is dependent on a system called the WZY. And it consists of 3 parts. The first is the WZA protein, which is a pore on the outer membrane. The second is WZC, which is a tyrosine autokinase in the inner membrane. And the third is WZB, which is a tyrosine phosphatase, which works with WZC, and it can also be nonspecific and functions in the cytoplasm. So a previous study found if you delete WZY, you can have an increased resistance to macrolide antibiotics. And another study found that if you delete only WZA, you can get the same phenotype. But no one has really discovered what WZB's role is. So one way that antibiotic resistance can happen is by phosphorylation. So on the left you have a dephosphorylated macrolide, and that is in its active form. So if a kinase comes by and phosphorylates it inactivates the macrolide. And vice versa, phosphatase can dephosphorylate it and return the macrolide to its active form. So this phosphatase function is what WZB does. So we thought if you get rid of WZB, maybe you can have more phosphorylated and inactivated macrolides that can lead to antibiotic resistance. Which led us to a research question: is deletion of WZB sufficient to confer resistance of E. coli K30 to macrolides? This helped form our hypothesis that the loss of function of WZB, a component of the E. coli K30 WZY capsule polymerization system, will result in increased capsule-independent macrolide resistance because WZB is a nonspecific cytoplasmic tyrosine phosphatase. So to investigate this, we used 5 strains. One of them was the WZA single knockout, another was the WZA and WZB double knockout, also the WZY single knockout, as well as, of course, the WZB single knockout, and also the wild type strain, which has the entire WZY cassette. So we took these 5 strains and did a PCR and DNA agarose gel to confirm the identity of the strains. Then we did a disc diffusion assay with erythromycin to test for the antibiotic resistance of each strain. And finally, we did an MICB assay also with erythromycin to determine the minimum inhibitory concentration of each strain. So the first question is, in the model about macrolide resistance, your group mentioned that phosphorylation of macrolides caused them to become inactive. Why is it thought that phosphorylation results in inactivation of the drug? So the way that macrolides work is it inhibits translation during protein synthesis. So it binds to this 50S subunit and prevents the translocation of the mRNA. So why does macrolide phosphorylation change anything? Well, it changes the structure. So if you see here, this is the structure of erythromycin, the macrolide that we worked with, and pay attention to this 2' hydroxyl group. If you phosphorylate erythromycin, then you lose that reactive 2' hydroxyl group. So referring back to this diagram, getting rid of that reactive hydroxyl inhibits macrolide binding to the 23S rRNA target site, which then restores proper translation and antibiotic resistance.
The next question that we're going to investigate is whether other factors, such as growth conditions or incubation temperature, could be contributing to the different results in our MICB assay and disc diffusion assay. We completed our disc diffusion on all of our experimental strains, and as seen from the graph on the left, the WZB mutant CWG344 was the only strain that displayed erythromycin sensitivity to the same extent as the wild-type strain E69. All other strains, including CWG655, CWG281, CWG343, displayed resistance to the erythromycin discs. We expected our MIC data to support the data that we generated from our disk diffusion. However, as seen from the table on the right, not all strains agree with the MIC data. For example, CWG281 shows resistance in the disk diffusion, but is sensitive in the MIC, with an MIC value of 100 micrograms per mil, which is the same as a wild-type E69. The same discrepancy was observed by the group who investigated the WZA knockout last term. Interestingly, the other strains show consistent results, with CWG655 and CWG343 displaying resistance in both assays, and CWG344 displaying sensitivity. This begs the question then of why the discrepancy is only seen between the WZA knockout strain. Some of the potential explanations for this observed discrepancy are as follows. First, the strains that are being plated on the solid plates may produce more capsule proteins when compared to liquid media, which has been previously identified in studies involving Staphylococcus aureus. In this case, although the WZA mutant is lacking the transmembrane channel to transport the capsular polysaccharides to the surface, it could still be producing more capsule in the solid media if the absence of WZA has led to an upregulation of a different protein or pathway that is involved in production of capsule. This could definitely be an area of research to investigate in future experiments. The next possible explanation is if the strains had different growth rates, and although they were diluted to the same OD before plating, the fact that they were in different phases of growth could have given them different susceptibilities to the antibiotics, which typically affect growing cells. Lastly, our peers asked whether differences in incubation time or temperature between the two assays could be contributing to the observed discrepancies. We can confidently say that the time and temperature were kept consistent between the two assays, and thus would not likely be an important factor in our observed discrepancy.
This next question comes from 1-alpha asking why we chose to investigate resistance to macrolides, specifically erythromycin, in our study, and whether there are previous evidence in literature to show there was resistance to the antibiotic. That is a great question, 1-alpha. We are more than glad to explain our reasoning behind investigating macrolides. Our initial interest in macrolide resistance involving the WZY system stems from a GEMI paper published in the 19th volume by Botros et al. Originally, Botros et al. Wanted to look at group 1 capsule's ability to induce antibiotic resistance in bacteria. Since capsule is used by bacteria to protect themselves from environmental stress, they were curious to see whether it was able to protect bacteria from antibiotics. Previous findings were inconclusive and inconsistent, so they decided to test it for themselves. They wanted to see the difference between wild-type K30 E. coli versus E. coli that lacked the capsule synthesis component in the WZY cassette. They looked at the wild-type E69 strain versus the WZY knockout CWG665 strain and their resistance to the variety of different antibiotic classes. They only observed differences in tetracycline and erythromycin sensitivity between the two strains. Since erythromycin is a common representative of macrolides, they decided to investigate other variants of the antibiotic. They observed that same resistance in the knockout to all the alternate macrolides. However, they did not find evidence to suggest that this was due to the lack of capsule formation in the knockout, and concluded that it must be the lack of the WZY proteins that caused the phenotype, which is why we decided to narrow in and see which protein was responsible for this resistance towards macrolides, using erythromycin as our common representative of the antibiotic class.
Here is our last question. Our last question came from Group 2 Ada, asking us how we can improve on our PCR reactions and prove that the faint band in the gel is indeed not an artifact. Well, this is another great question, and luckily we have an answer for it. In the original PCR reaction, we claimed that the faint band, as indicated with the red box, is a PCR result of CWG655 delta strain. The reason why we believe this is not just a simple artifact is based on its comparison with the previous report. The size of the band is consistent with the result produced by Su et al., who previously have used the same primers and the same PCR protocol. However, the intensity of the band is not optimal, and we have decided to repeat the experiment with few modifications, hoping to improve on our PCR qualities. Here is the picture of our PCR reactions on a gel with few modifications in our experimental protocols. There were total 3 changes we made in an attempt to improve on our PCR qualities. First of all, we used the brand new aliquoted primers in which have not been previously thawed. Repeated thawing and freezing have been documented to have a negative impact on the primer viability. Secondly, we increased the amount of DNA used in each reactions. Lastly, we repeated the experiment with dimethyl sulfoxide, also known as DMSO. DMSO is a known enhancer for PCR as it prevents the formation of secondary structure in DNA templates. As indicated in the figure above, more bands have successfully been shown. Also, the band intensity increases with the addition of DMSO. Overall, there is an improvement in the PCR quality, and we can conclude our modifications enhanced our PCR reactions. Furthermore, we were able to successfully reproduce a band consistent with our previous PCR result, indicating that this band is the expected band for CWG 655 delta strain.